Good evening and welcome to Dreamy Academy. Tonight we drift far back through time and also incredibly close to a scale of existence that hums just beyond the reach of our everyday senses. As we begin, I'd love to know where you are watching from, your city, your country, perhaps even what the night sky looks like outside your window. If you enjoy these peaceful explorations of science, please take a moment to like the video and subscribe. It truly helps this little corner of quiet curiosity grow. And for those already subscribed, thank you sincerely for being part of this slow and thoughtful adventure. You are the quiet constellation that guides these nightly journeys. Right now, as you listen, forces older than mountains are shaping the atoms in your bones, and processes more ancient than the first star are unfolding within the cells of your body. We often look to the vastness of the cosmos for wonder, to the swirling galaxies and the deep, silent spaces between them. And indeed, wonder resides there, boundless and humbling. But there is another universe, equally vast in its implications, equally ancient in its origins, and profoundly intricate in its design, that exists not out there, but all around us, and even within us. It is a world teeming with life, a tapestry woven from threads too fine for the eye to see, yet it underpins every breath you take, every bite you eat, every landscape you admire. Tonight, we will begin to explore this hidden realm, the world of microbiology. It is a journey not into emptiness, but into a fullness beyond imagining, a place where life first learned to be, where the fundamental work of existence has been carried out for billions of years, and where the most profound truths about life on Earth are whispered in the language of molecules and single cells. This isn't a world of distant stars, but of intimate, immediate, and ceaseless activity. It is a wonder that, when contemplated with quiet patience, can lull the mind into a state of profound peace, a gentle surrender to the sheer elegance of existence itself. So let us begin our quiet drift into the unseen. Have you ever paused to consider the air you breathe, the soil beneath your feet, or even the surface of your own skin? To our unaided senses, these seem like familiar, perhaps even mundane spaces. The air feels empty, the soil appears inert, and our skin a protective barrier. Yet, beneath this visible surface of the world, a vibrant, bustling, and ancient civilization thrives. It is a civilization of the unimaginably small, a world where life exists in forms so tiny that 1,000,000 of them could dance on the head of a pin, yet so numerous and powerful that they have, for billions of years, been the true architects of our planet. There's something beautiful happening in every speck of dust, every drop of dew, every ripple of ocean water. It is the silent, ceaseless work of the microscopic, the realm of the microbial. These are not just tiny creatures. They are the fundamental operators of Earth's grand systems, the recyclers of elements, the producers of oxygen, the quiet partners in every living thing. To understand them, we must first learn to stretch our imagination, not outwards to the stars, but inwards, into scales that challenge our everyday perception, into a world beneath the threshold of our seeing eyes, yet vibrant with purpose. To truly appreciate this unseen world, we must first try to grasp its scale. Imagine, if you will, shrinking down not just a little, but profoundly. If you could shrink to the size where a grain of sand felt like a mountain, you would still be too large to truly perceive the microbial world. A human hair, which seems so fine to us, would become a colossal tree trunk, and a single human cell, the basic unit of our own bodies, would appear as a sprawling city, complex and intricate, with highways and factories and communication networks. But even a human cell is, in many ways, an entire continent compared to the inhabitants of the microbial realm. Most bacteria, for instance, are measured in micrometers. A micrometer, or micron, is 1/1,000,000th of a meter. To put that into perspective, the average human hair is about 100 micrometers thick, so a bacterium might be 1/100th the thickness of a single strand of your hair. Some viruses are even smaller, measured in nanometers, 1/1000,000th of a meter. This is a scale so tiny that the very concept of seeing, as we understand it, with light reflecting off surfaces, begins to break down. It is a world of such profound minuteness that our senses simply evolved to ignore it. Yet, it is precisely at this scale, where matter and energy interact in the most fundamental ways, that life first sparked and continues to thrive. Each bacterium, each archaeon, each tiny fungus, each virus is a universe of molecular machinery, perfectly adapted to its niche, carrying out the intricate dances of biochemistry with an efficiency that humbles even our most advanced technologies. This is where the true stories of life's endurance, adaptability, and boundless creativity unfold, silently, patiently, endlessly. For most of human history, this entire world remained utterly unknown to us. We lived our lives, breathed our air, tilled our soil, and battled our illnesses, completely unaware of the invisible forces at play. It was only with the invention of the microscope, a device that bent light to reveal what was hidden, that this secret universe began to unveil itself. It was a Dutch draper, Antony van Leeuwenhoek, in the late 17th century, who first peered through his meticulously ground lenses and saw what he called animalcules, tiny wriggling creatures in a drop of water, in scrapings from his teeth, even in pepper infusions. He was seeing bacteria, protozoa, and other single-celled organisms for the very first time, sketching them with an astonished precision that still resonates today. Leeuwenhoek's discovery was a quiet revolution, revealing that life was not merely what we could see, but an immense, unseen ocean teeming with forms and functions beyond our wildest imaginings. What he saw was not an anomaly, but the norm. Everywhere he looked, life was there, dancing and striving, and what we have learned since then, over centuries of patient observation and rigorous experimentation, is even more astonishing. This microbial world is not just present, it is ubiquitous. It is everywhere. Consider for a moment the soil beneath a single tree. A single teaspoon of healthy soil can contain more microorganisms, bacteria, fungi, archaea, protozoa, than there are people on Earth. Think of that density of life, a universe of activity in such a tiny volume. Or a single drop of ocean water, a seemingly clear bead on your finger, might hold thousands of bacteria and viruses, each a distinct entity, each playing its role in the vast interconnected web of marine life. Even your own body, which you might think of as a singular entity, is in fact a walking, breathing, thriving ecosystem. Trillions of microbial cells, collectively known as your microbiome, reside on your skin, in your gut, and in every accessible nook and cranny, outnumbering your own human cells by a factor of 10 to 1. They are not invaders, they are partners, silent participants in your health, your digestion, and even aspects of your mood. These invisible architects are the quiet engines of our planet. They cycle nutrients, breaking down dead matter, and returning essential elements like nitrogen and carbon to the living world. They produce the very oxygen we breathe, with ancient cyanobacteria having transformed Earth's early atmosphere, making complex life possible. They decompose pollutants, shape the fertility of our soil, and even influence global weather patterns. Without them, the intricate dance of life on Earth would grind to a halt. There would be no fertile soil, no breathable air, no cycles of decay and renewal. We are not just surrounded by them, We are utterly dependent on their tireless, unseen work. The beauty in this profound interconnectedness is a humbling thought. We, with our grand cities and complex technologies, are but recent arrivals, passengers on a planet long governed by these tiny ancient beings. Our very existence is a testament to their enduring success, their ability to transform and sustain the Earth through billions of years of geological and atmospheric change. They are the quiet foundation, the ceaseless hum beneath the symphony of macroscopic life. This deep dive into the microbial scale reveals not just smaller versions of what we already know, but entirely different ways of being alive. It shows us that life is far more resilient, diverse, and interconnected than our everyday perception suggests. It is a world of constant adaptation, of biochemical artistry, where the simplest forms can achieve the most profound effects. There is a particular comfort in understanding that even in the smallest corners of existence there is order, purpose, and an astonishing vibrancy. So we've begun to glimpse the vastness and the ubiquity of the microscopic world, a realm that exists just beyond our vision, yet underpins everything we experience. We've seen how tiny these architects are, and how utterly essential their silent work is to the planet and to ourselves. But what are these myriad tiny entities really? What is the fundamental unit of life at this incredible scale, and how did it first come into being? That question leads us into the very heart of life's design, into the elegant simplicity and astonishing complexity of the cell itself. Welcome back, dear friends, to the tranquil halls of the Dreamy Academy. In our inaugural chapter, we embarked upon a journey across the vast hidden landscapes of the microbial world, marvelling at the ubiquitous presence of the invisible architects that quietly shape our planet. We considered the profound implications of their ancient lineage and their ceaseless work, from the soil beneath our feet to the very air we breathe, and even within the intricate ecosystems of our own bodies. Yet as we concluded our initial exploration, a deeper, more fundamental question lingered, a quiet whisper in the vastness of the unseen. What truly is this tiny entity that we call a microorganism? What is its core design, its essential blueprint? Today, we delve into the very heart of this mystery, journeying inward, past the scale of a human hair and a grain of sand, to uncover the foundational unit of all life, the cell. Imagine, if you will, the most intricate, self-sustaining miniature world, bustling with purpose, organized with an efficiency that defies our grandest architectural feats. This is the cell, the atom of biology, the single irreducible vessel that holds the very essence of existence. Long before Antony van Leeuwenhoek peered through his lenses and beheld his animalcules, the cell had been theorized, glimpsed, and finally undeniably recognized as the fundamental building block. It was a gradual revelation, a tapestry woven by many hands across centuries. We owe much to the patient observations of pioneers like Robert Hooke, who in the 17th century first coined the term cell after observing the honeycomb-like structures in a thin slice of cork. He saw empty chambers like the cells of a monastery, and the name endured, though he could not have known the vibrant living universe contained within. Or in the 19th century, with improved microscopy, the true significance began to unfold. Matthias Schleiden, a botanist, declared that all plants are composed of cells, followed swiftly by Theodor Schwann, a zoologist, who extended this principle to animals. Finally, Rudolf Virchow articulated the profound truth that all cells arise from pre-existing cells. Together, these insights coalesced into what we now revere as cell theory, a cornerstone of biology, affirming that the cell is the basic unit of life, and that life begets life through this miraculous microscopic form. So let us now step through an imaginary threshold, shrinking our perspective to truly embrace the cellular scale. As we do, we discover that the world of cells is not monolithic. Rather, it reveals two grand architectural designs, two ancient lineages that have diverged and flourished across billions of years. We first encounter the prokaryotic cell, the venerable elder of life's family tree. These are the bacteria and archaea, the original invisible architects, whose simple yet profoundly effective design has allowed them to thrive in every conceivable niche on Earth, from the crushing depths of the ocean to the searing heat of volcanic vents, and even within the very fabric of our being. A prokaryotic cell is a marvel of minimalist efficiency. Picture, if you will, a tiny self-contained workshop, perhaps a bustling single-room studio where all the essential work of life is carried out without internal partitions. Its primary defining feature is its sheer simplicity. There is no nucleus, no elaborate network of internal membranes dividing functions. Instead, its precious genetic material, its complete blueprint, typically a single circular strand of DNA, floats freely within the cytoplasm, concentrated in a region we call the nucleoid. This DNA is the master plan, dictating the construction and operation of the entire cell. Encasing the vibrant interior is the cold cell membrane, a flexible, intelligent barrier, a sentry that meticulously regulates what enters and exits the cell, maintaining its delicate internal environment. Outside this membrane, many prokaryotes possess a rigid cell wall, a sturdy outer fortification that provides structural support and protection, much like the ancient walls of a citadel guarding its precious contents. Some, like a ship outfitted for movement, may even sport whip-like flagella, allowing them to propel themselves through their aqueous worlds, or tiny hair-like pili for attachment to surfaces. And scattered throughout the cytoplasm, like countless industrious little machines, are the ribosomes, the cell's protein factories, tirelessly translating the genetic code into the proteins that perform almost every function, from building structures to catalyzing reactions. This seemingly unassuming design, this single-room workshop, is not primitive in its function, but rather a testament to elegant engineering. Prokaryotes are incredibly adaptable, quick to reproduce, and astonishingly diverse in their metabolic capabilities. They are the true masters of resilience, having pioneered many of the biochemical pathways that underpin all life on Earth. They existed for billions of years as the sole inhabitants of our planet, quietly terraforming it, preparing the stage for all that was to follow. Then, approximately 2 billion years ago, a monumental evolutionary leap occurred. From the ancient simplicity of the prokaryote emerged a new, more complex form, the eukaryotic cell. This was not merely an upgrade, but a profound reimagining of cellular architecture, akin to evolving from a single-room studio to a sprawling multi-story mansion, complete with specialized wings and dedicated departments. The most striking feature of the eukaryotic cell is its compartmentalization. Imagine a bustling city where distinct districts are dedicated to specific tasks: a central library and control room, power plants, manufacturing facilities, recycling centers, and transportation networks. This is the eukaryotic cell. Its most prominent feature, the very definition of its name, EU, meaning true, karyon meaning nucleus, is the nucleus. Here, encased within its own double-membraned envelope, lies the cell's genetic library, its DNA, organized into multiple linear chromosomes. The nucleus is the cell's control center, safeguarding the precious blueprint and regulating the expression of genes. orchestrating the entire cellular symphony. But the complexity does not end there. The eukaryotic cell is replete with membrane-bound organelles, each a miniature organ performing a specific vital function. The mitochondria, often called the powerhouses of the cell, are like tiny energy plants tirelessly converting nutrients into ATP, the cell's currency of energy. In plant cells and some protists, we find chloroplasts, the green architects of life, capturing sunlight and transforming it into chemical energy through the miraculous process of photosynthesis, a direct echo of the ancient prokaryotic cyanobacteria that first oxygenated our planet. Beyond these energy converters, a vast network of internal membranes crisscrosses the cytoplasm, the endoplasmic reticulum, ER. A labyrinthine system of interconnected sacs and tubules serves as the cell's manufacturing and transport hub, synthesizing proteins and lipids. Proteins destined for secretion or insertion into membranes are folded and modified here. Adjacent to the ER, the Golgi apparatus, often compared to the cell's post office or packaging plant, further processes, sorts and packages these molecules into vesicles for delivery to their final destinations. Then there are lysosomes, the cell's recycling and waste disposal units, breaking down cellular debris and foreign invaders with potent enzymes, and within the cytoplasm, a dynamic cytoskeleton. A network of protein filaments provides structural support, facilitates movement, and acts as a highway system for transporting vesicles and organelles. The evolution of such intricate compartmentalization was a revolutionary step. It allowed eukaryotic cells to grow much larger than their prokaryotic ancestors, to perform a wider array of functions simultaneously, and to achieve a level of specialization that underpins the diversity of multicellular life. It meant that a cell could be both a power plant and a manufacturing hub, a library and a recycling center, all operating efficiently within its own boundaries. Yet despite these profound architectural differences, the simplicity of the prokaryote versus the elaborate complexity of the eukaryote, a deep resonant truth binds them together, the universal blueprint of life. Both cell types share fundamental molecular machinery. Both rely on DNA as their genetic material, RNA to carry messages, and ribosomes to synthesize proteins. Both are enveloped by a cell membrane composed of a lipid bilayer. This shared heritage speaks volumes, pointing unequivocally to a common ancestor, a primordial cell from which all life, in its astonishing variety, has descended. It is a testament to the enduring power of evolution, building upon foundational designs, refining and diversifying them over eons. As we contemplate this intricate design, this fundamental unit of life, we are inevitably drawn back to the profound mystery that concluded our last chapter, the origin of the cell itself. How did such an organized, self-replicating entity first arise from the primordial soup of early Earth? How did the first cell membrane form, encapsulating the nascent molecular machinery that would begin the dance of life? While the exact pathways remain subjects of intense scientific inquiry and wonder, we understand that these first cells were likely prokaryotic in nature, simple yet robust, capable of harnessing energy and replicating their genetic information. They were the original spark, the first iteration of the invisible architects, laying the very foundations upon which all subsequent life would build. To truly appreciate the cell is to recognize it not as a static blueprint, but as a vibrant, dynamic entity, a universe of molecular machinery in constant, purposeful motion. Within its microscopic confines, molecules collide and bind, enzymes catalyze reactions with breathtaking speed, Genetic information is faithfully copied and expressed, and structures are continually built, repaired, and recycled. It is an orchestral symphony of biochemical processes, playing out ceaselessly, billions of times over, in every living being every moment. This inner world, this bustling cellular city, is the stage upon which the grand drama of life unfolds. From the simplest bacterium to the most complex neuron in your brain, the cell is the irreducible unit of vitality, a silent testament to the genius of natural design. It is a profound realization, is it not, that within each of us and all around us exist these miniature worlds, operating with an ancient wisdom and an unparalleled efficiency. As we move forward, we will continue to explore the intricate functions of these cellular wonders, delving into how they communicate, how they divide, and how they collectively create the breathtaking diversity of life we see. But for now, let us simply sit with the wonder of the cell, the fundamental blueprint, the true beginning of all things seen and unseen. The genesis of the first cell, a singular epoch-making event, remains one of the grandest mysteries, a whispered question at the very dawn of life. How did the primordial soup, a chaotic brew of simple molecules, energized by lightning strikes, volcanic heat, and the sun's unfiltered ultraviolet radiation, coalesce into that first self-replicating, membrane-bound entity? It is a puzzle that continues to inspire awe and relentless inquiry, a profound riddle that challenges our understanding of complexity from simplicity. We stand at the precipice of this enigma, gazing back billions of years, humbled by the sheer improbability of that initial spark. Yet, while we ponder that ultimate origin, a mystery perhaps forever beyond our full grasp, we can turn our gaze to the incredible, tangible legacy of those earliest pioneers, the direct descendants of that very first cellular spark, the prokaryotes. These are the ancient ones, the true enduring architects of our living world, whose fundamental blueprints were drawn billions of years ago. For too long, perhaps, we have spoken of them as being simple in their cellular architecture, often contrasting their lack of a nucleus and other membrane-bound organelles with the intricate compartmentalization of their eukaryotic cousins. But to call them merely simple risks profoundly understating their sophistication, their breathtaking adaptability, and the sheer, unyielding power of their collective existence. It is a misnomer that diminishes their monumental contributions. Perhaps instead we should view their structure not as a lack, a deficiency in complexity, but as an elegant, minimalist design honed over eons of relentless natural selection. It is a design perfectly suited for rapid reproduction, efficient resource utilization, and unparalleled survival, enabling them to dominate across every conceivable niche on Earth. Their simplicity is, in fact, a testament to optimized efficiency, a profound example of doing more with less, a masterclass in biological engineering. Imagine, if you will, stepping into a bustling metropolis where every inhabitant, though microscopic, performs a vital, intricate task with unwavering precision. This is the unseen world of prokaryotes, a vibrant, bustling and ancient civilization, a universe of molecular machinery operating with unparalleled efficiency, a silent symphony of biochemical processes. They are not merely uniform blobs, indistinct and undifferentiated. Their forms are as varied and purposeful as the functions they perform, each shape a strategic adaptation. Some are perfect spheres, like tiny iridescent pearls or meticulously crafted marbles, which we call cocci. often clumping together in grape-like clusters or long chains. Others stretch into rod-like cylinders, bacilli, resembling microscopic grains of rice or slender capsules, designed for maximizing surface area for nutrient absorption or efficient movement through viscous fluids. Then there are the spirilla, gracefully coiled like miniature corkscrews or elegant ribbons, their helical shape allowing them to bore through dense matrices or move with a distinctive undulating swim. And the vibrios, shaped like elegant commas or crescent moons, often found in aquatic environments, their curved bodies aiding in directional motility. Each morphology, a testament to countless generations of evolutionary fine-tuning, offers specific advantages in movement, attachment to surfaces, nutrient uptake, or evasion of predators, a silent declaration of form following function. But their true marvel lies not just in their shapes, but in their metabolic ingenuity, a biochemical wizardry that predates and indeed shaped all subsequent life. If the first cells were born into a world devoid of oxygen, a planet simmering with volcanic activity and primordial oceans, what incredible biochemical pathways must have evolved to harness energy from the raw elemental forces of the young Earth. Prokaryotes are the undisputed masters of this unseen alchemy. They are the great transformers, the quiet alchemists who have, for billions of years, orchestrated the planet's most fundamental chemical cycles, turning the seemingly inert into the vibrant and life-sustaining. They unlocked the secrets of energy conversion long before the sun's energy could be directly utilized for complex life, pioneering every metabolic trick in the book. Consider the autotrophs among them, the self-feeders, those remarkable organisms capable of producing their own food. Some, like the cyanobacteria, are photosynthetic, much like plants, capturing the sun's energy with ancient pigments to convert atmospheric carbon dioxide into organic matter. And crucially, in this process, they release the oxygen that would eventually flood our atmosphere, a slow monumental transformation that began over 2.5 billion years ago. They are the ancient breath givers, the silent sculptors of the very air we inhale, turning a toxic gas for early life forms into the lifeblood of aerobic existence. Imagine the sheer scale of this planetary engineering, a single-celled revolution that changed the course of life forever. Others are chemosynthetic, a truly astonishing feat of biochemical innovation. They do not rely on sunlight at all, but instead draw energy from inorganic chemical reactions, oxidizing compounds like ammonia, sulfur, iron, or even hydrogen sulfide. Picture these microscopic beings thriving in the lightless depths of hydrothermal vents, where superheated mineral-rich water spews from the Earth's crust, or within the very bedrock of our planet, miles beneath the surface sustained by geological processes. They create vibrant ecosystems where no light can penetrate, proving that life's ingenuity knows no bounds, finding sustenance in the most unlikely and extreme corners of existence. Then there are the heterotrophs, the countless species that consume organic matter, acting as the planet's ultimate recyclers. They are the tireless decomposers, breaking down dead organisms, fallen leaves, discarded waste products, and the molecular detritus of life, returning vital nutrients like nitrogen, phosphorus, and carbon to the soil and water. Without their ceaseless, tireless work, Earth would be a barren wasteland, choked with undecomposed organic debris, a planet suffocating under its own spent biological matter. They are the silent custodians of decomposition, the essential cleaners of our global ecosystem, ensuring the perpetual flow of life's building blocks, a never-ending cycle of renewal. This metabolic diversity is the engine behind their ubiquity and their incredible adaptability. Where can you find prokaryotes? The more pertinent question might be, where can't you find them? They thrive in environments that would instantly extinguish most other forms of life, often pushing the very boundaries of what we consider habitable. We call them extremophiles for good reason, and their existence forces us to redefine the parameters of life itself. Some revel in scorching hot springs existing at temperatures far exceeding the boiling point of water, their enzymes perfectly stable and active where human proteins would instantly denature. Others flourish in environments of extreme cold, nestled within polar ice caps, beneath glaciers, or deep ocean sediments, their metabolism slowed but persistent. Still others call home the crushing pressures of the abyssal plains, where the weight of miles of ocean water is unimaginable, or the searing acidity of volcanic lakes, where pH levels plummet to near zero. Conversely, some thrive in the caustic alkalinity of soda lakes, while others can withstand intense radiation, surviving doses that would sterilize a human, or live in hypersaline solutions, far saltier than any ocean. These are often the archaea, a distinct domain of prokaryotes, the true champions of these harsh realms, reminding us that life's tenacity is a force beyond our easy comprehension, a relentless drive to persist and proliferate. They are in the soil beneath your feet, a single gram teeming with billions, a microscopic jungle of intertwined life, each particle a world unto itself. They populate every drop of water, from the purest mountain spring to the most polluted urban river, silently processing nutrients and waste. They drift invisibly in the air you breathe, carried by currents, settling on every surface, microscopic travellers on global winds. And most intimately they reside within us. For we are not solitary beings, we are walking ecosystems, complex symphonies of interacting life forms, a grand consortium of cells. Within your gut, on your skin, in your mouth, lives a vast and intricate community of microorganisms, your personal microbiome. These are your quiet partners, often engaging in mutualistic relationships, performing functions essential for your health that your own cells cannot. They help digest complex carbohydrates and fibers that our enzymes cannot break down, synthesize vital vitamins like K and some B vitamins, train your immune system to distinguish friend from foe, and even influence your mood and cognitive function through the gut-brain axis. They are so profoundly integrated into our biology, so interwoven with our very existence, that it becomes impossible to consider ourselves without them. This intricate dance of coexistence honed over millions of years highlights that life at its most fundamental is about collaboration, a shared journey of survival and flourishing. The sheer numbers are staggering. The collective biomass of prokaryotes on Earth is immense, dwarfing that of all plants and animals combined. They represent the vast majority of Earth's biodiversity, a hidden realm of countless species, many yet undiscovered, each contributing to the intricate tapestry of life. They are the fundamental operators, the silent engine driving the planet's geochemistry, regulating its climate, shaping the very conditions that allow larger, multicellular life forms, like us, to exist and thrive. Without them, the intricate web of life would unravel, and our planet would be an entirely different, inhospitable place. As we journey through the Dreamy Academy, we continue to peel back the layers of the unseen to understand the foundational principles of life. We began with the cell, the universal unit, then distinguished the two great domains of cellular life. Now, we have taken a closer look at the ancient, enduring power of the prokaryotes, the bacteria and archaea, revealing their astonishing diversity, their unparalleled adaptability, and their indispensable roles in every corner of our world and even within our own bodies. They are not just the past, they are the present and the future, constantly evolving, constantly shaping. In the chapters ahead, we will delve deeper into the specific wonders of these microscopic marvels, exploring the intricate mechanisms that govern their lives and the profound ways they continue to shape our planet. For the story of microbiology is, in essence, the story of life itself, told from its most ancient, fundamental, and awe-inspiring perspective. We have journeyed together through the ancient, elegant simplicity of the prokaryotic world, marveling at its boundless ingenuity and the sheer resilience of life's first true architects. We've seen how these tiny, single-celled wonders, devoid of internal partitions, have mastered every conceivable niche, shaping our planet for eons. But as we stand at the threshold of Chapter 4, our gaze shifts gently toward a profound transformation, a leap in complexity that would fundamentally redefine the very fabric of life. Imagine, if you will, the primordial Earth, a swirling cauldron of chemical possibility. For billions of years, prokaryotes reigned supreme, their metabolic pathways weaving the intricate biochemical tapestry of the nascent biosphere. Then a quiet revolution began, not with a roar, but with an embrace, a profound symbiotic dance that would give rise to a new kind of cell, one capable of an astonishing level of internal organization and specialization. We are now invited into the realm of the eukaryotic cell, a universe of molecular machinery far more intricate, yet born from the very simplicity we have just explored. The defining narrative of the eukaryotic cell is often encapsulated in a theory so elegant, so profoundly beautiful, that it feels less like scientific hypothesis and more like an ancient epic poem. Endosymbiosis. Picture a larger, ancient prokaryotic cell, perhaps an archaeon, drifting through the primordial soup. It encounters a smaller, agile bacterium, one particularly adept at converting oxygen into energy, a nascent mitochondrion. In a moment of evolutionary serendipity, the larger cell engulfs the smaller, but instead of digesting it, a remarkable partnership forms. The smaller cell finds a safe haven, a steady supply of nutrients, while the host gains an unparalleled advantage. A dedicated, highly efficient energy generator. This was not a fleeting encounter, but a permanent inherited bond, a fusion of destinies that would echo through all subsequent life. This pivotal event, this ancient cellular embrace, was not a singular occurrence. Later, in a separate, equally momentous act of cellular symbiosis, certain eukaryotic cells, the ancestors of plants and algae, also engulfed another type of bacterium, a photosynthetic one, brimming with the ability to harness sunlight. These became the chloroplasts, the verdant engines of our planet, transforming light into life-sustaining sugars. The evidence for this extraordinary tale is woven into the very fabric of these organelles. Mitochondria and chloroplasts retain their own circular DNA, strikingly similar to bacterial DNA. They divide independently, much like bacteria, and their internal membranes bear the unmistakable hallmarks of their prokaryotic ancestry. They are, in essence, ancient bacterial guests, domesticated and integrated, still humming with the echoes of their free-living past, now indispensable powerhouses within a larger, more complex cellular architecture. With this grand narrative of endosymbiosis in mind, we can now truly appreciate the defining characteristics of the eukaryotic cell. Unlike the elegant simplicity of prokaryotes, The eukaryotic cell is a sprawling city, bustling with specialized districts, each with its own vital function. Its most distinguishing feature, the very origin of its name, eukaryon meaning true nucleus, is the nucleus itself. This grand membrane-bound chamber houses the cell's precious genetic material, its complete library of instructions meticulously organized into chromosomes. Here, within this central command center, the complex symphony of gene expression is orchestrated, dictating the identity and function of the entire cell. Imagine a vast ancient library, its scrolls carefully catalogued, its wisdom guarded and accessed with precision that is the nucleus, the brain and heart of the eukaryotic cell. Beyond the nucleus, the eukaryotic cell is a marvel of internal compartmentalization, a sophisticated network of membrane-bound organelles, each performing a specialized task, allowing for an extraordinary division of labor. These internal membranes create distinct environments, optimizing biochemical reactions and greatly increasing the cell's overall efficiency and capacity. Let us wander through this cellular metropolis. Mitochondria. As we've discussed, these are the indispensable powerhouses of the cell, tirelessly converting nutrients into adenosine triphosphate, ATP, the universal energy currency of life. Their folded inner membranes, called cristae, dramatically increase the surface area for these vital energy-generating reactions, a testament to their ancient, highly efficient bacterial design. Endoplasmic reticulum, ER. This vast interconnected network of membranes forms a cellular highway, snaking throughout the cytoplasm. The rough ER, studded with ribosomes, is the primary site for the synthesis and initial processing of proteins destined for secretion or insertion into membranes. The smooth ER cell, devoid of ribosomes, is involved in lipid synthesis, detoxification of drugs and poisons, and calcium storage. It is the cell's intricate manufacturing and transport system. Golgi apparatus, often described as the cell's post office, the Golgi apparatus is a stack of flattened membrane-bound sacs called cisternae. Here, proteins and lipids synthesized in the ER are further modified, sorted, and packaged into vesicles for transport to their final destinations, whether within the cell or exported outside. It is the meticulous sorter and shipper, ensuring every molecular package reaches its correct address. Lysosomes and peroxisomes. These are the cell's recycling and detoxification centers. Lysosomes, abundant in animal cells, contain powerful digestive enzymes that break down waste materials, cellular debris, and even invading bacteria. Peroxisomes specialize in breaking down fatty acids and detoxifying harmful substances, often producing hydrogen peroxide as a byproduct, which they then neutralize. They are the vigilant custodians, maintaining cellular cleanliness and safety. Vacuoles. Particularly prominent in plant cells, a large central vacuole can occupy up to 90% of the cell volume, storing water, nutrients, and waste products, and maintaining turgor pressure against the cell wall. In animal cells, smaller vacuoles and vesicles perform various storage and transport functions. They are the cell's versatile storage units and pressure regulators. Cytoskeleton. Providing structural support, facilitating movement, and organizing organelles, the cytoskeleton is a dynamic network of protein filaments, microfilaments, intermediate filaments, and microtubules, crisscrossing the cytoplasm. It is the cell's internal scaffolding, its railway system for transport, and its muscular system for shape changes and locomotion. The sheer scale of this internal organization allows eukaryotic cells to be significantly larger and more complex than their prokaryotic ancestors. While a typical bacterium might be 1 to 5 micrometers in diameter, a eukaryotic cell can range from 10 to 100 micrometers, sometimes even larger. This increase in size and complexity opened up entirely new evolutionary avenues, paving the way for multicellularity and the breathtaking diversity of life we see around us. And what a diversity it is! When we think of eukaryotes, our minds often jump immediately to the familiar macroscopic worlds of animals and plants. But these are just two branches of a much larger, more ancient and incredibly diverse eukaryotic tree. Let us not forget the vast, often unseen kingdoms of single-celled and colonial eukaryotes. Protists. This is perhaps the most astonishingly diverse group, a catch-all kingdom for eukaryotes that are not animals, plants, or fungi. From the graceful whip-like flagellates that propel themselves through water, to the ever-changing forms of amoebas that engulf their prey, to the intricate glass-shell diatoms that form the base of many aquatic food webs, protists are a testament to eukaryotic adaptability. They can be photosynthetic, like algae, producers of much of Earth's oxygen, heterotrophic, like protozoa consuming other microbes, or even parasitic causing diseases. They are the true microbial shapeshifters, often overlooked but ecologically indispensable. Their beauty, visible under the microscope with their intricate cilia, flagella or pseudopods, is a silent symphony of form and function. Fungi. These remarkable organisms, from the microscopic yeasts that leaven our bread to the sprawling mycelial networks beneath our feet, represent another distinct eukaryotic lineage. Unlike plants, they are heterotrophic, obtaining nutrients by secreting digestive enzymes onto their food source and absorbing the broken-down molecules. Their cell walls are made of chitin, a unique polysaccharide, and they play crucial roles as decomposers, nutrient recyclers, and symbiotic partners with plants. Plants and animals. These are the macroscopic eukaryotes we most readily recognize, the culmination of billions of years of cellular evolution and the epitome of multicellular complexity. Yet every leaf, every feather, every beating heart is ultimately composed of these fundamental eukaryotic cells, working in concert, each performing its specialized role within the grand symphony of an organism. As we ponder the eukaryotic cell, we are struck once again by the profound interconnectedness of all life. Even the most complex multicellular organism, built from countless eukaryotic cells, still hosts and relies upon a vast and vital community of prokaryotes, our microbiome, for instance, a testament to the enduring ancient partnerships forged billions of years ago. The eukaryotic cell, with its internal complexity and its capacity for multicellularity, represents a monumental evolutionary achievement, a testament to life's ceaseless drive towards innovation and adaptation. We have now journeyed from the raw simplicity of the earliest self-replicating cells to the elegant efficiency of prokaryotes, and finally to the breathtaking complexity and internal architecture of eukaryotes. We've seen how life, through profound symbiotic unions, built ever more intricate structures, allowing for an astonishing array of forms and functions. Yet despite their differences, a common thread runs through all these cellular designs, the universal molecular blueprint, the fundamental chemical language shared by every living thing. As we conclude this chapter, reflecting on the intricate wonders of the eukaryotic cell, we carry with us an even deeper appreciation for the invisible architects, not just the prokaryotes, but also their eukaryotic descendants, from the humble amoeba to the towering redwood. Each cell, a universe in miniature, silently performing its duties, contributing to the vast, living tapestry of our world. In our next exploration, we will delve into the very essence of life's continuity, how these amazing cells reproduce, ensuring the endless dance of existence continues. It is another quiet marvel awaiting our gentle inquiry. Welcome back, my dear companions, on this quiet journey, to the tranquil halls of the Dreamy Academy. We have journeyed through the vast, unseen landscapes of cells, from the ancient, elegant simplicity of prokaryotes to the wondrous, compartmentalized complexity of eukaryotes. We have marveled at their structures, their evolutionary dances, and their profound roles as the very architects of our living world. But now we must venture even deeper, beyond the visible boundaries of membranes and organelles, into a realm of exquisite detail where the true magic of life unfolds, the molecular tapestry woven from threads too fine for any optical lens to discern. Imagine, if you will, that we have shrunk once more, not merely to the size of a cell, but to a scale where even the most intricate organelle appears as a sprawling, silent city. Within this city, however, is a ceaseless, vibrant symphony of motion, a universe of molecular machinery engaged in an unending, purposeful dance. This is where the fundamental essence of life truly resides, in the ceaseless interactions of molecules, tiny, intricate structures that, when combined and orchestrated with breathtaking precision, give rise to all that we know as living. As we stand at this threshold, gazing into the molecular heart of existence, a profound reverend horn settles upon us. It is here, at this infinitesimal scale, that the universal language of life is spoken, a language older than mountains, shared by every bacterium, every plant, every animal, and indeed by every one of us. We are about to explore the primary molecular players, the four grand classes of macromolecules, each a masterpiece of nature's design, collaborating to sculpt, sustain, and replicate life itself. These are the proteins, the carbohydrates, the lipids, and the nucleic acids, the very bedrock upon which all cellular function is built. Let us begin with the proteins. The tireless workhorses, the versatile artisans of the cell. If a cell were a bustling factory, proteins would be its diverse workforce, the structural beams that hold it together, the assembly line workers, the transport vehicles, the security guards, and the specialized engineers. They are, in essence, the executors of life's countless tasks. Each protein is a marvel of three-dimensional architecture, meticulously folded into a unique shape that dictates its specific function. Consider the sheer diversity. Some form the rigid scaffolding of the cell, like the cytoskeleton we glimpsed in eukaryotes. Others are tiny motors, propelling bacteria or moving cargo within cells. Still others are receptors, listening to the cell's environment, or antibodies defending against invaders. But perhaps their most astonishing role is as enzymes. These are the molecular catalysts speeding up biochemical reactions by millions, even billions, of times without being consumed themselves. Without enzymes, the metabolic process is essential for life, breaking down food, synthesizing new molecules, generating energy, would proceed at a glacial pace, far too slow to sustain any living organism. They are the patient, efficient facilitators, orchestrating the countless chemical transformations that define metabolism. Each enzyme is exquisitely specific, like a master key fitting only one lock, ensuring that the right reactions occur at the right time. They are, quite literally, the facilitators of life's ceaseless flow. At their core, proteins are polymers, long chains built from smaller repeating units called amino acids. There are 20 common types of amino acids, and the sequence in which they are linked, like letters forming a word, determines the protein's unique three-dimensional shape and, consequently, its function. A single misstep in this sequence, a misplaced letter, can alter the protein's delicate folding, rendering it dysfunctional, a poignant reminder of the precision required for life to flourish. We are, in a very real sense, walking, breathing collections of these molecular machines, each performing its assigned duty with quiet dedication. Next, we turn our gaze to the carbohydrates, the primary energy providers and structural supporters of the living world. Think of them as the immediate fuel and the sturdy scaffolding. From the simplest sugars to the most complex starches and fibers, carbohydrates are fundamental. The sweet taste of fruit, the stored energy in a potato, the rigid structure of a tree trunk, all owe their existence to these versatile molecules. The most basic units are the monosaccharides, or simple sugars such as glucose. Glucose is the universal energy currency, the immediate fuel for cellular respiration, powering the very existence of both prokaryotic and eukaryotic cells. It is the swift, accessible energy source that keeps the molecular machinery humming. These simple sugars can then be linked together to form longer chains, or some polysaccharides. Some polysaccharides, like starch in plants or glycogen in animals, serve as long-term energy storage, reservoirs of fuel that can be tapped when immediate glucose is scarce. Others, like cellulose, which forms the strong cell walls of plants, or chitin, which gives structure to fungi and insect exoskeletons, provide crucial structural support. These are the sturdy beams and protective shields of the molecular world. The elegance of carbohydrates lies in their ability to be both rapidly accessible energy and enduring structural components. They are the swift currents and the unyielding shores of the cellular ocean, indispensable for the myriad forms of life that populate our planet. Now, let us consider the lipids. Often misunderstood, yet profoundly essential molecules. These are the wise guardians, the dense energy reserves, and the crucial messengers. What unites lipids is their defining characteristic. They are largely hydrophobic, meaning they fear water and do not dissolve in it. This seemingly simple property is fundamental to life. The most iconic lipids are the phospholipids, the silent architects of every cellular membrane. Imagine a double layer of these molecules, their water-fearing tails pointing inward, away from the watery environments inside and outside the cell, and their water-loving heads facing outward. This arrangement creates a selective barrier, a delicate but robust wall that defines the cell's boundaries, controlling what enters and exits. It is the very skin of the cell, allowing the internal environment to be distinct from the external, a crucial step in the evolution of life. Without this phospholipid bilayer, cells as we know them simply could not exist. Beyond membranes, lipids serve as highly efficient energy storage molecules, far denser than carbohydrates. Fats, for instance, are packed with energy, providing insulation and cushioning. Other lipids, like steroids, act as vital signaling molecules, carrying messages throughout complex organisms, influencing growth, development, and metabolism. They are the quiet, often unseen orchestrators of cellular identity and communication. Finally, we arrive at the sole nucleic acids, the venerable keepers of information, the master blueprints of life itself. These are the molecules that store, transmit, and express the genetic instructions that make each organism unique, yet undeniably linked to all others. The most famous of these is DNA, deoxyribonucleic acid. The grand library of genetic information, meticulously coiled into the iconic double helix. Think of DNA as an ancient, incredibly detailed scroll, containing the complete instructions for building and operating an organism, passed down through countless generations. Each strand of DNA is a polymer, a long chain composed of repeating units called nucleotides. Each nucleotide has 3 components: a sugar, a phosphate group, and a nitrogenous base. It is the precise sequence of these bases, adenine, guanine, cytosine, and thymine, along the DNA strand that forms the genetic code, the language of inheritance. This code dictates the sequence of amino acids in proteins, thus indirectly controlling every aspect of cellular structure and function. Then there is RNA, ribonucleic acid. DNA's versatile cousin. RNA acts as the messenger, carrying copies of genetic instructions from the DNA in the nucleus, in eukaryotes, to the protein-making machinery in the cytoplasm. It also plays crucial roles in regulating gene expression, and even possesses enzymatic activity itself. RNA is the dynamic interpreter, the diligent scribe, translating the timeless wisdom of DNA into the active commands that build proteins. And let us not forget ATP, adenosine triphosphate, a specialized nucleotide that serves as the primary energy currency of the cell. While not a long-term information storage molecule like DNA, ATP is the immediate universal fuel that powers nearly every cellular process, from muscle contraction to active transport across membranes. It is the spark, the instant energy packet that keeps the molecular symphony playing. As we reflect on these 4 classes of macromolecules, proteins, carbohydrates, lipids, and nucleic acids, a profound truth emerges. They do not exist in isolation. They are interwoven, interdependent, engaged in a ceaseless, intricate dance. Proteins work with lipids to form membranes. Carbohydrates are attached to proteins and lipids on cell surfaces for recognition. Nucleic acids provide the instructions for making all the proteins. This dynamic interplay, this constant collaboration, is the very essence of life's molecular machinery. It is in the microscopic realm where molecules collide, bind, and transform with such astonishing precision that the universe of molecular machinery truly manifests. These universal building blocks are the threads from which the rich tapestry of all life, from the simplest archaegon thriving in a volcanic vent to the most complex human being, is woven. They are the testament to a shared ancestry, a common blueprint that whispers across billions of years of evolution. We have begun to peel back another layer, moving from the grand architecture of cells to the intricate designs of their molecular components. This journey, my friends, is far from over. Having understood what these molecules are, our next quiet exploration will delve into how they are used, how cells harness energy, how they grow, how they divide, and how they communicate. For in understanding these fundamental molecular processes, we truly begin to grasp the enduring wonder and silent power of the invisible architects that shape our world. Until then, let us carry the image of this molecular ballet within us, a reminder of the profound complexity that underpins every moment of existence. Having journeyed through the intricate architecture of the cell, and admired the molecular tapestry woven from proteins, carbohydrates, lipids, and nucleic acids, we now arrive at a profound question. How does this magnificent machinery move? What ignites its ceaseless activity, powers its growth, and sustains its very existence? The answer lies in the dynamic, vibrant heart of every living entity: metabolism. Imagine, if you will, a sprawling ancient city, built not of stone and steel, but of molecular components, each with a purpose. This city is alive, constantly rebuilding, repairing, and responding to its environment. Its streets hum with activity. Goods are transported, structures are erected, and energy is generated to fuel every single process. This bustling metropolis is a single cell, and its metabolism is the sum total of all the chemical reactions that occur within it, transforming matter and energy to sustain life. It is an astonishingly complex, yet perfectly orchestrated symphony of reactions, a testament to billions of years of silent, efficient evolution. We perceive the world around us as largely static, yet beneath the surface, within every leaf, every drop of water, every grain of soil, and indeed, within every cell of your own body, a ceaseless transformation is underway. Metabolism is the grand conductor of this molecular orchestra, ensuring that every note, every reaction, plays its part in harmony. It is the very essence of what it means to be alive, the continuous ebb and flow of energy and matter that defines the boundary between the animate and the inanimate. At its core, metabolism can be divided into two fundamental yet complementary processes: catabolism and anabolism. Picture them as two sides of the same exquisitely balanced coin, constantly flipping and turning to maintain the delicate equilibrium of life. Catabolism is the art of deconstruction, the breaking down of larger, complex molecules into simpler ones. Think of it as dismantling an old building to reuse its bricks and beams, or perhaps more aptly, as harvesting the energy stored within the fuel we consume. When a cell breaks down glucose, for example, it is engaging in catabolism, releasing the stored chemical energy from its bonds. This energy, once liberated, is then captured and transformed into a usable form, much like converting raw fuel into electricity. Catabolic reactions are often exergonic, meaning they release energy and they provide the raw materials for the cell's construction projects. Conversely, anabolism is the process of construction, the building up of complex molecules from simpler precursors. If catabolism is demolition, anabolism is architectural design and construction. It's the synthesis of new proteins from amino acids, the creation of new strands of DNA, or the assembly of complex carbohydrates. These reactions are typically endogonic, meaning they require an input of energy, much like building a house requires effort and resources. Anabolic processes are vital for growth, repair, and the storage of energy. The beauty of metabolism lies in how these two seemingly opposing forces are meticulously intertwined, with the energy released from catabolism directly fueling the energy-demanding processes of anabolism. But how, you might wonder, is this energy actually stored and transferred within the cell? What is the universal currency that every cellular transaction accepts? Here, we introduce you to one of life's most elegant and ubiquitous molecules, adenosine triphosphate, or ATP. Imagine ATP as the cell's rechargeable battery, or perhaps a tiny, perfectly designed energy token. Structurally, ATP is a marvel of efficiency. It consists of an adenine base, a ribose sugar, and 3 phosphate groups linked together. The magic, the true power, resides in the bonds between these phosphate groups, particularly the terminal one. These are often referred to as high-energy phosphate bonds. When a cell needs energy for an anabolic reaction, to synthesize a protein, to move a muscle, to transport a molecule across a membrane, it simply spends an ATP molecule. This involves breaking the bond between the second and third phosphate groups, releasing a burst of usable energy and transforming ATP into ADP, adenosine diphosphate, and an inorganic phosphate group. The beauty of this system is its reversibility. Once the energy has been expended, ADP can be recharged back into ATP by adding a phosphate group, a process that requires energy input, typically derived from catabolic reactions. This constant cycle of ATP hydrolysis, release of energy, and ATP synthesis, recharge, is the engine that drives life. It is the rhythmic pulse that keeps the cellular city vibrant, ensuring that energy is always available precisely when and where it is needed, without wasteful excess. This intricate dance of breaking and building, of energy release and capture, would be impossibly slow and inefficient without the help of nature's most extraordinary catalysts, enzymes. We briefly touched upon proteins in our last chapter, and here we see their unparalleled versatility in action. Most enzymes are proteins, exquisitely folded into precise three-dimensional shapes, each designed to facilitate a specific biochemical reaction. Consider an enzyme as a highly skilled artisan, possessing the perfect tools and knowledge to transform raw materials into a finished product, or to dismantle a complex structure with remarkable speed and precision. Without the artisan, the process might still occur, but it would be agonizingly slow, perhaps taking millennia instead of milliseconds, or requiring extreme conditions that would be lethal to the delicate cell. Enzymes work by lowering the activation energy of a reaction, the initial energy barrier that must be overcome for a reaction to proceed. They do not get consumed in the reaction, nor do they change the overall energy balance. They simply make the impossible, or at least the impractical, possible and efficient. Their specificity is profound. Like a master key designed for one particular lock, each enzyme typically acts on only one or a few specific molecules, called substrates, guiding them through their chemical transformations with unparalleled accuracy. With ATP as the energy currency and enzymes as the diligent facilitators, cells are equipped to extract energy from their environment. For many organisms, particularly the vast majority of microorganisms we are exploring, the primary source of energy comes from breaking down organic molecules, most notably glucose. This journey of glucose breakdown is a central pillar of catabolism and can unfold in a few remarkable ways. One of the most ancient and universal pathways is glycolysis, a series of 10 enzymatic reactions that occur in the cytoplasm of virtually all living cells, from the simplest bacterium to the most complex human cell. Glycolysis literally means sugar splitting, and it does just that. It breaks down a 6-carbon glucose molecule into 2 3-carbon pyruvate molecules, yielding a small net gain of ATP and some electron carriers. It is a testament to the common ancestry of life that this pathway has persisted largely unchanged for billions of years. A foundational energy harvesting strategy that predates the oxygen-rich atmosphere we know today. From pyruvate, the metabolic path diverges, depending largely on the presence or absence of oxygen. In the presence of oxygen, many organisms, including us, proceed to cellular respiration. A highly efficient process that extracts a tremendous amount of energy from pyruvate. This multi-stage pathway, involving the Krebs cycle and the electron transport chain, occurs in the mitochondria of eukaryotic cells and in the cytoplasm and across the cell membrane of prokaryotic cells. It is here that the vast majority of ATP is generated through a process akin to a molecular waterfall, where electrons are passed down a chain of protein complexes, releasing energy that is used to pump protons and ultimately drive ATP synthesis. This is the powerhouse of the cell, the ultimate expression of aerobic energy generation. However, not all life forms and not all environments are blessed with abundant oxygen. In its absence, many microorganisms resort to fermentation. This anaerobic pathway is less efficient in terms of ATP yield compared to aerobic respiration, but it allows cells to continue generating some ATP from glucose by regenerating the necessary electron carriers for glycolysis. There are numerous types of fermentation, each producing different end products. Lactic acid in our muscles during intense exercise, ethanol in yeast used for brewing and baking, or acetic acid in vinegar production. These diverse fermentative pathways highlight the incredible metabolic flexibility of microorganisms, enabling them to thrive in oxygen-depleted niches across the planet. But what about the organisms that don't rely on breaking down pre-existing organic molecules for energy? How do they begin this grand metabolic dance? Here we turn our gaze to the autotrophs, the self-feeders, who stand at the very foundation of most ecosystems. Among them, photosynthesis is the most renowned. This miraculous process, performed by plants, algae, and a vast array of photosynthetic bacteria, like the cyanobacteria we encountered earlier, captures the radiant energy of sunlight and converts it into chemical energy in the form of glucose. It is the original energy transformation, the cosmic breath that fills the atmosphere with oxygen and provides the organic molecules that fuel nearly all other life on Earth. Photosynthesis is an anabolic triumph, using light energy to build complex sugars from simple carbon dioxide and water, literally transforming light into life. And even beyond sunlight, some remarkable microorganisms, the chemoautotrophs, have mastered an entirely different art of energy capture. Thriving in the darkest, most extreme environments, hydrothermal vents at the bottom of the ocean, deep within the Earth's crust, or in highly acidic hot springs. These invisible architects harness the chemical energy stored in inorganic molecules like hydrogen sulfide, ammonia, or iron. They oxidize these compounds, releasing energy to synthesize their own organic molecules, entirely independent of sunlight. This metabolic ingenuity reveals the astonishing adaptability of life, proving that where there is a chemical gradient, there is a potential for life to flourish. As we reflect on these diverse metabolic pathways, we are struck by their profound interconnectedness. The waste products of one organism often become the vital nutrients for another. The oxygen produced by photosynthetic organisms is consumed by aerobic respirers. The organic matter generated by autotrophs fuels the vast catabolic endeavors of heterotrophs. This intricate web of energy flow and nutrient cycling is the very heartbeat of our planet. A continuous dynamic exchange orchestrated largely by the invisible architects we are studying. From the simplicity of a single glucose molecule being broken down, to the majestic capture of solar energy, to the harnessing of geothermal chemical reactions, metabolism is the universal engine. It is a testament to the elegant efficiency of life, a constant quiet hum of activity that sustains every breath, every thought, every movement, and indeed, every microscopic vibration of existence. We are all, in our own ways, participants in this grand metabolic story, a story that began billions of years ago with the first cells and continues today, ceaselessly, all around us, and even within us. Our journey through the Dreamy Academy continues to unfold the layers of life's mysteries. Having understood how cells manage their energy, our next exploration will delve into the fascinating world of how they grow, how they reproduce, and how they pass on their genetic blueprint from one generation to the next, ensuring the unbroken continuity of life itself. The stage is set for the incredible saga of cellular growth and division. The magnificent metabolic dance we explored in our last gathering, the ceaseless breaking down and building up, the intricate pathways of energy flowing through every living cell, all of it serves a grander, more profound purpose. It is not merely a cycle for its own sake, but the very fuel for life's most fundamental imperative, the perpetuation of its own existence. Today, dear students of the Dreamy Academy, we turn our gaze to the breathtaking processes by which these tiny, vibrant worlds grow, divide, and meticulously pass on the sacred blueprint of life to the next generation. Imagine, if you will, the bustling city of a single cell. Every metabolic reaction, every enzyme's swift work, every molecule of ATP generated contributes to the expansion of this city. For a microorganism, growth is not merely an increase in size, though that is often part of it. It is a profound and coordinated accumulation of all its components, proteins, lipids, carbohydrates, and, most crucially, its precious genetic material. It is a quiet, internal expansion, a preparation for the ultimate act of self-renewal. And then, the moment arrives when growth culminates in division. It is a moment of singular importance, a testament to life's enduring resilience and its relentless drive to continue to multiply, to weave its intricate tapestry across time and space. We are about to witness the miraculous mechanisms by which these invisible architects, from the simplest prokaryote to the most complex eukaryote, ensure their legacy. Let us begin with the elegant simplicity of the prokaryotic world, the realm of bacteria and archaea. Their method of reproduction, known as binary fission, is a marvel of efficiency and speed, a testament to their minimalist yet potent design. Imagine a single-celled organism, a tiny self-contained universe quietly expanding. Its internal components double, its membrane stretches, and its singular circular chromosome, that coiled thread of destiny, begins its own meticulous duplication. This process begins with the replication of the DNA. The double helix, that elegant spiral staircase of genetic information, gently unzips, much like a zipper opening down its length. Along each separated strand, new complementary nucleotides are precisely laid down, guided by the original template. The result is two identical copies of the entire genome, two perfect blueprints where there was once only one. This is not a casual copying, it is an act of exquisite precision, ensuring that the genetic inheritance is faithfully passed on. Once the DNA has been duplicated, the cell continues to grow, elongating as if stretching towards an unseen horizon. The two identical chromosomes, now distinct entities, migrate to opposite ends of the expanding cell. In the middle, a new cell wall and cell membrane begin to form, pinching inward, creating a dividing partition or septum. This septum grows progressively inward until it completely separates the parent cell into two distinct genetically identical daughter cells. It is a gentle parting, a quiet fission, yet it is the engine of their astounding proliferation. Think of it. A single bacterium, under ideal conditions, can divide every 20 minutes. This exponential growth is why prokaryotes are such formidable forces in our world, capable of colonizing new environments with breathtaking speed, adapting, evolving, and shaping ecosystems on a global scale. Their simplicity, far from being a limitation, is their strength, allowing for rapid reproduction and unparalleled adaptability. Binary fission is not just a biological process. It is a fundamental principle of their enduring power, a quiet, ceaseless hum of life replicating itself billions of times over, all around us, and even within us. But what of the eukaryotic cells, those architects of complexity that house their genetic treasures within a true nucleus and boast an array of specialized organelles? Their journey of reproduction is a more elaborate, highly orchestrated ballet, a fascinating sequence of events that ensures the precise distribution of their far more extensive genetic material. This process for asexual reproduction is called mitosis, and it is a cornerstone of growth, repair and renewal in multicellular organisms, as well as the primary mode of reproduction for many single-celled eukaryotes like protists and yeasts. Before a eukaryotic cell can embark on the dramatic performance of mitosis, it must first prepare. It enters a preparatory phase known as interphase, a period of intense activity where the cell grows, carries out its normal metabolic functions, and, crucially, duplicates its entire complement of DNA. Just as with the prokaryotes, this DNA replication is a meticulously supervised process, ensuring that each of the many chromosomes is copied perfectly, resulting in two identical sister chromatids joined together. This is the S phase, synthesis of the cell cycle, nestled between two gap phases, G1 and G2, where the cell grows and checks for errors. With its genetic material doubled and its internal machinery ready, the cell then enters the mitotic phase. Imagine a grand stage being set, spotlights dimming, and the principal players, the chromosomes, preparing for their intricate dance. The first act is prophase. The long, diffuse threads of DNA, known as chromatin, begin to condense, coiling and folding upon themselves until they become visible under a microscope as compact, X-shaped chromosomes. Each X represents two identical sister chromatids, still tethered at a central point. Simultaneously, the nuclear envelope, the protective membrane surrounding the nucleus, begins to dissolve, gracefully receding like a curtain rising. Meanwhile, tiny protein structures called centrosomes begin to move to opposite poles of the cell, unfurling a network of protein fibers, the spindle fibers, which will act as the guiding ropes and pulleys for the chromosomes. Next comes metaphase, perhaps the most visually striking part of the performance. The condensed chromosomes, guided by the spindle fibers, meticulously align themselves along the cell's equator, forming a perfectly straight line known as the metaphase plate. It is a moment of exquisite order, a precise arrangement that ensures each daughter cell will receive a complete and balanced set of genetic instructions. Each chromosome is now poised, awaiting its cue. The third act, anaphase, is a sudden, dramatic separation. The sister chromatids, which have been bound together, are abruptly pulled apart by the shortening spindle fibers. Each chromatid, now considered a full-fledged chromosome, is drawn swiftly and gracefully towards opposite poles of the cell. It is a synchronized exodus, a perfectly coordinated migration, ensuring that identical sets of genetic material are delivered to what will become the two new cells. The cell elongates further, stretching to accommodate this internal division. Finally, telophase brings the performance to a close. As the chromosomes arrive at their respective poles, they begin to decondense, unfurling back into their more diffuse chromatin state. New nuclear envelopes form around each set of chromosomes, re-establishing two distinct nuclei within the single elongated cell. the spindle fibers disappear, their work complete. Simultaneously, cytokinesis. The division of the cytoplasm usually begins, pinching the cell in two from the middle, much like the septum formation in binary fission. The result is two genetically identical daughter cells, each a perfect replica of the parent. This intricate dance of mitosis, with its precise choreography and meticulous execution, underscores the complexity and elegance of eukaryotic life. It is slower, more energy-intensive than binary fission, but it allows for the accurate segregation of vast amounts of genetic information, critical for the development and maintenance of complex multicellular organisms. From the healing of a wound to the growth of a tree, mitosis is the silent, ceaseless engine of renewal, a testament to the enduring power of cellular continuity. Underlying both binary fission and mitosis, providing the very foundation for the faithful transmission of life, is the miraculous process of DNA replication. We touched upon it briefly in the context of prokaryotes, but its significance warrants a deeper contemplation. DNA, that magnificent double helix, is not merely a molecule, it is the ultimate blueprint, the sacred text, encoding every instruction, every possibility, every nuance of an organism. To pass on life, this blueprint must be copied perfectly and precisely. Imagine the double helix, a ladder twisted into a spiral. In replication, specialized enzymes act as molecular unzippers, breaking the weak bonds between the complementary base pairs, adenine with thymine, guanine with cytosine, that form the rungs of the ladder. The two strands separate, each now serving as a template. Then other molecular architects, the DNA polymerases, move along each exposed strand, meticulously adding new, free-floating nucleotides from the cellular environment. A with T, G with C, the rules are absolute, ensuring perfect complementarity. This process is known as semi-conservative replication. Each new double helix is not entirely new. It consists of one original parental strand and one newly synthesized daughter strand. It is a brilliant design, a built-in mechanism for accuracy, ensuring that the genetic information is not only copied, but also conserved across generations. Imagine copying a priceless ancient manuscript, but instead of making a new copy from scratch, you carefully peel apart the original pages, and then, for each original page, you create a perfect new companion page. Each new book thus contains half the original and half new. The fidelity of this process is astounding. While errors or mutations can occur, the cell has its own sophisticated proofreading and repair mechanisms, constantly scanning and correcting any missteps. This precision is vital, for even a tiny alteration in the genetic code can have profound consequences. It is this unwavering commitment to accurate replication that ensures the stability of species, while the rare, uncorrected mutation provides the raw material for evolution, allowing life to adapt and diversify over eons. As we reflect on these processes, the quiet unfolding of binary fission, the intricate ballet of mitosis, and the meticulous precision of DNA replication, we are once again humbled by the invisible architects. They have devised mechanisms of such elegance and efficiency that they ensure the continuity of life itself, from the simplest bacterium thriving in the most extreme environments to the complex cells that form our very being. This journey through cell growth and reproduction reveals not just the mechanics of life's perpetuation, but also its profound determination. Every division, every replication, is a statement of existence, a quiet assertion that life will continue, will thrive, will adapt. We, In our dreamy academy, are privileged to witness these unseen engines of continuity, to marvel at the microscopic precision that underpins the macroscopic world, and to understand that the legacy of life is woven strand by meticulous strand, generation after generation, in an endless, beautiful cycle. We carry these blueprints within us, and they are the echoes of billions of years of ceaseless, wondrous reproduction. Our next steps will lead us into the fascinating world of genetic expression, exploring how these blueprints are read and translated into the very fabric of life. As we journey deeper into the intricate cosmos of the cell, we have marveled at its architectural designs, its tireless energy engines, and the elegant choreography of its reproduction. We have witnessed the DNA blueprint meticulously copied, ensuring the faithful transmission of life's instructions from one generation to the next. Yet, a profound question still whispers in the quiet corners of our minds. How does this inert double helical molecule, this sacred scroll of genetic information, actually become life? How do the sequences of its nucleotides translate into the vibrant dynamic reality of a functioning cell, into the very fabric of being? This is where our exploration takes a breathtaking turn, inviting us to witness the ultimate act of creation within every living entity. We are about to uncover the exquisite molecular dance that reads the silent language of DNA and translates it into the bustling activity of proteins, the true workhorses, the architects and engineers, the very essence of cellular function. It is a process so fundamental, so universal, that it underpins every breath taken, every thought conceived, every quiet bloom of a microorganism in the soil. Picture, if you will, the DNA as the ancient, invaluable manuscript stored in the grand library of the cell's nucleus, or the central archives of a prokaryote's nucleoid. This manuscript holds all the wisdom, all the instructions for building and operating the entire cellular civilization. But this precious original cannot simply be taken out and used on the chaotic construction sites of the cell. It must be protected, preserved. So a meticulous scribe is employed, tasked with creating temporary single-use, working copies of specific sections of this blueprint. This act of copying, this sacred transcription, is our first stop. It is a process known as transcription, where the genetic information encoded in a segment of DNA is faithfully transferred into a molecule of ribonucleic acid, or RNA. Think of RNA as DNA's versatile cousin. While DNA is the sturdy double-stranded master blueprint, RNA is typically single-stranded, more flexible, and contains the sugar ribose instead of deoxyribose, and the nucleotide uracil U in place of thymine, T. This subtle difference makes RNA perfectly suited for its role as a temporary messenger and an active participant in protein synthesis. The star enzyme orchestrating this copying is polymerase, a molecular artisan that glides along the DNA helix, unwinding a small section, and then, with breathtaking precision, synthesizes a complementary RNA strand. It reads the DNA sequence, a pairing with U instead of T, G with C, and assembles a new RNA molecule, growing nucleotide by nucleotide. Once a specific gene or section of the blueprint has been copied, the RNA polymerase detaches and the newly formed RNA molecule is released. In the complex eukaryotic cell, this transcription takes place safely within the nucleus, protecting the DNA. The newly synthesized RNA, often called messenger RNA, mRNA, then undergoes some sophisticated processing, like a rough draft being refined and edited before it's ready for publication. In the more streamlined prokaryotic cell, transcription occurs in the cytoplasm, often simultaneously with the next stage, a testament to their elegant efficiency. But mRNA is not the only type of RNA involved in this grand molecular ballet. There are other crucial players. Transfer RNA, tRNA, which we will meet shortly, acts as a molecular ferry carrying specific building blocks, and ribosomal RNA, rRNA, which forms the structural and catalytic core of the cellular protein factories. Each type of RNA plays its indispensable role in bringing the DNA's silent instructions to life. Once the mRNA copy is complete and, if necessary, processed, it embarks on its journey from the nucleus to the bustling construction zones of the cytoplasm. Here, the true magic of translation begins, the conversion of the mRNA's nucleotide sequence into the amino acid sequence of a protein. Before we witness this translation, we must first understand the language itself. The genetic information is not read letter by letter, but in words of 3 nucleotides called codons. Each codon specifies a particular set amino acid, the fundamental building block of proteins. For example, the RNA sequence AUG is a codon that signals for the amino acid methionine and also serves as a start signal for protein synthesis. UAA, UAG, and UGA are stop codons signaling the end of the protein chain. What is truly astonishing, truly humbling, is that this genetic code is nearly universal across all known life forms on Earth. From the simplest bacterium to the towering redwood, from the deepest ocean microbe to the human brain, the codon AUG always means methionine, and UAA always means stop. This profound universality is one of the most compelling pieces of evidence for the common ancestry of all life, a shared ancient language spoken by every living cell that has ever existed. It is a whisper from the deep past, binding us all in an unbroken chain of molecular communication. Now, with our mRNA blueprint in hand and an understanding of the genetic code, we arrive at the bustling construction site, the ribosome. These intricate cellular machines, composed of RNA and proteins, are the factories where proteins are synthesized. Imagine them as tiny, highly sophisticated assembly lines, waiting to receive their instructions and begin their work. The process of translation unfolds in 3 main stages: initiation, elongation, and termination. In initiation, the mRNA molecule first docks with a small ribosomal subunit. Like a conductor preparing the orchestra, this subunit scans the mRNA until it finds the start codon, AUG. Then, a special tRNA molecule carrying the amino acid methionine arrives and binds to this start codon. This tRNA is unique because it carries an anticodon, a 3-nucleotide sequence that is complementary to the mRNA codon. The large ribosomal subunit then joins, completing the functional ribosome, and the assembly line is ready to roll. Next comes elongation, the heart of protein synthesis. The ribosome moves along the mRNA, reading one codon at a time. As each new codon enters the ribosome's A site, a corresponding tRNA molecule, carrying its specific amino acid, arrives and binds to it via its complementary anticodon. The amino acid carried by the incoming tRNA is then joined to the growing chain of amino acids by a peptide bond, forming the nascent protein. The ribosome then shifts, moving the now empty tRNA out and making space for the next one, allowing the polypeptide chain to lengthen progressively. This process is like a series of molecular handoffs, each tRNA delivering its precious cargo, precisely guided by the mRNA instructions. This continues, codon after codon, until the ribosome encounters a stop codon on the mRNA. This signals REGE termination. There are no tRNAs that correspond to stop codons. Instead, special release factors bind to the stop codon, triggering the release of the newly synthesized polypeptide chain from the ribosome. The ribosome then disassembles, freeing the mRNA to potentially be translated again or to be degraded, its message now delivered. The newly formed polypeptide chain, a string of amino acids, is not yet a functional protein. Like a raw material, it must now fold into a precise, intricate three-dimensional shape. This folding is crucial, as a protein's function is dictated by its structure. Some proteins fold spontaneously, guided by the interactions between their amino acids, while others require the assistance of molecular chaperones. Once folded, these proteins can then embark on their diverse roles. As enzymes catalyzing reactions, as structural components providing shape and support, as transporters moving molecules, as signals communicating between cells, or as defenders protecting against invaders. Indeed, the vast array of functions we explored in Chapter 5, from the intricate dance of metabolism to the very scaffolding of the cell, are all carried out by these magnificently crafted proteins. It is a profound realization. From the silent wisdom of DNA, transcribed into the transient message of mRNA, and then translated into the dynamic structures of proteins, the very essence of life emerges. This continuous flow of information from gene to protein is the central dogma of molecular biology, a guiding principle that illuminates how the blueprint becomes being, how potential becomes reality. But the story does not end with a protein being made. Cells are not mindless factories churning out every protein indiscriminately. Instead, they are master orchestrators controlling when and how much of each protein is produced. This is the realm of gene expression regulation, a sophisticated system that ensures cells only make the proteins they need, precisely when and where they are needed. Imagine a vast orchestra with thousands of instruments, but only a select few play at any given moment, guided by the conductor's subtle cues. Similarly, within a cell, various molecular conductors determine which genes are expressed, transcribed and translated, and which remain silent. This regulation allows cells to adapt to changing environments, to differentiate into specialized types in multicellular organisms, and to maintain the delicate balance of homeostasis. In the world of our invisible architects, this level of control is paramount. A bacterium might quickly activate genes to break down a new food source that has appeared in its environment, or a yeast cell might switch on genes for fermentation when oxygen becomes scarce. These subtle yet powerful regulatory mechanisms allow microorganisms to exhibit their astonishing adaptability and resilience, thriving in conditions that would be lethal to more complex life forms. As we conclude this chapter, reflect on the immense journey we have undertaken. We began with the invisible world, scaled down to the cell, then to its molecular machinery, its energy pathways, and its methods of reproduction. Now, we have witnessed the ultimate transformation, how the coded information of DNA is brought to life through the elegant processes of transcription and translation, culminating in the creation of proteins that perform virtually every function within the cell. This ceaseless, precise, and breathtaking molecular dance is happening within every cell of your body, within every bacterium floating in the air, within every protozoan swimming in a pond. It is a testament to the enduring power and exquisite ingenuity of life at its most fundamental level. The whispers of life carried by mRNA and translated by ribosomes, truly build the world we know, shaping the invisible architects and indeed shaping us. We are all woven from these same molecular threads, bound by this universal language, forever connected to the ancient, profound wisdom encoded within a simple double helix. Our next steps will lead us to explore how these microscopic entities interact with their environments and with each other, how they adapt and evolve, further revealing the dynamic tapestry of the microbial world. As we journey deeper into the intricate tapestry of life woven from threads too fine for the unaided eye, we have come to understand the language of existence, the genetic code, transcribed and translated into the tireless proteins that orchestrate every cellular function. We have marveled at the precision with which life replicates its blueprint, ensuring continuity. But what if the blueprint itself could change? What whispers of alteration, what profound shifts allow life to adapt, to flourish in ever new forms, to dance to the rhythm of an evolving world? Today, dear explorer, we turn our gaze to the dynamic heart of life's perpetual motion, genetic variation, and evolution. For the universe of molecular machinery is not static. It is a vibrant, ceaselessly adapting symphony, where the notes themselves can be rewritten, leading to entirely new melodies of existence. We will uncover the mechanisms by which these changes occur, shaping the vast diversity of microorganisms that have sculpted our planet for eons. Imagine, if you will, the DNA strand as an ancient, infinitely long scroll, penned with the instructions for a cell's entire being. Most of the time, this scroll is copied with exquisite fidelity, but occasionally a scribe makes an error, a subtle misplacement of a letter, a skipped word, or an accidental addition. These are mutations. The fundamental wellspring of all genetic variation. They are often random, quiet events, whispers in the vast genetic library, yet they hold the power to reshape destinies. A single nucleotide substitution where one base is swapped for another, a T instead of a C for instance, can be like changing a single letter in a sentence. Sometimes this change is silent, like substituting a synonym that doesn't alter the meaning, quick for fast. The resulting protein remains unchanged, a silent testament to the redundancy within the genetic code. Other times it might lead to a missense mutation, where a different amino acid is incorporated, perhaps altering the protein's function subtly or even dramatically. Imagine changing ship to sheep, a small change with a large consequence for the journey. And then there are nonsense mutations, where a single change can prematurely terminate the protein synthesis, like a sudden, abrupt stop sign in the middle of our sentence, usually rendering the protein non-functional. More impactful still are insertions and deletions, where one or more nucleotides are added or removed from the sequence. These are often far more disruptive, causing a frameshift mutation. Think of reading a sentence in groups of 3 letters, like codons. If you remove or add a letter at the beginning, every subsequent 3-letter group shifts, changing the entire meaning from that point onward. This often leads to completely non-functional proteins, a dramatic rewriting of the cellular script. While many mutations are neutral, and some are deleterious, even lethal, it is the rare, beneficial mutation that serves as the raw material for natural selection. It is the chance improvement in the blueprint that allows an organism to thrive in a new environment, to resist a toxin, or to utilize a novel food source. These beneficial changes, though infrequent, are the gentle shapers of evolution, allowing life to continuously explore new possibilities. Yet for prokaryotes, The venerable elders of our planet, there is an even more direct and rapid pathway to genetic innovation, a means of sharing wisdom and adapting collectively. Horizontal gene transfer, HGT. Unlike the vertical transmission of genes from parent to offspring, HGT allows prokaryotes to exchange genetic material across species boundaries, even between distantly related organisms. It is a vast, invisible network of genetic sharing, an ancient internet of life, allowing for breathtakingly swift evolution and adaptation. Consider cell transformation, a process of elegant simplicity. Some bacteria possess the remarkable ability to actively take up naked DNA from their environment. This DNA might be released from dead cells, fragments of genetic wisdom floating free. A competent bacterium can internalize these fragments and, if they share sufficient homology with its own genome, integrate them. Imagine a cell drinking genetic insights from its surroundings, incorporating new instructions directly into its own blueprint. This is how a bacterium might acquire a gene for antibiotic resistance from a fallen comrade, or a new metabolic pathway, transforming its capabilities in a single, profound act. Then there is transduction, a surprising and intricate dance mediated by bacteriophages, the viruses that specifically infect bacteria. These phages are like tiny cosmic postal workers, inadvertently carrying genetic messages between bacterial cells. When a phage infects a bacterium, it hijacks the cell's machinery to produce more phages. Occasionally, during this frantic replication, fragments of the host bacterium's DNA are accidentally packaged into new phage particles instead of, or in addition to, the viral genome. When these transducing phages then infect another bacterium, they inject the acquired bacterial DNA, potentially transferring new genes to the recipient. It is a fascinating twist of fate where a predator becomes an unwitting courier of genetic exchange, accelerating the bacterial evolutionary journey. Finally, we encounter conjugation, often described as bacterial mating, though it is not a reproductive process in the eukaryotic sense. This is an intimate, direct transfer of genetic material from one bacterium to another through cell-to-cell contact. A donor bacterium, typically possessing a specialized piece of circular desine called a plasmid, often an F plasmid or fertility plasmid, extends a protein appendage called a pilus to a recipient cell. The pilus acts like a bridge, drawing the cells together. Through this bridge, a copy of the plasmid, or sometimes even a portion of the donor's main chromosome, is transferred. This is a powerful mechanism for spreading beneficial traits, such as antibiotic resistance genes or genes for virulence, rapidly through a bacterial population. Imagine a secret handshake across the microbial void, passing on vital survival skills. These mechanisms of horizontal gene transfer are not mere academic curiosities. They are foundational to the resilience and adaptability of microorganisms. They explain the rapid emergence of antibiotic resistance, the swift evolution of pathogens, and the incredible metabolic versatility of microbial communities. They underscore the interconnectedness of life on a scale far beyond our immediate perception, a constant sharing and remixing of genetic information that fuels the invisible architects of our world. As we ponder these dynamic processes, we are led to a deeper appreciation of the overarching organization of life itself. We have spoken of prokaryotes and eukaryotes, but molecular insights, particularly the comparison of ribosomal RNA, rRNA sequences, have unveiled a more profound division, revealing the three domains of life: bacteria, archaea, eukarya. This classification, pioneered by Carl Woese, revolutionized our understanding of evolutionary relationships, showing that life branched into three distinct lineages from a universal common ancestor. The primordial bacteria domain encompasses the vast majority of prokaryotes we typically encounter and study. They are the ubiquitous, diverse, and metabolically ingenious organisms we have come to know. The cyanobacteria that painted our skies with oxygen, the nitrogen-fixing bacteria enriching our soils, the decomposers recycling nutrients, and the teeming populations within our own bodies. They possess peptidoglycan cell walls, unique membrane lipids, and a remarkable array of metabolic pathways, continuing their ancient and essential work across every conceivable niche on Earth. The archaea domain, initially mistaken for bacteria, represents a lineage as ancient and distinct as bacteria themselves. While outwardly resembling bacteria in their prokaryotic cell structure, their molecular machinery, especially their ribosomal RNA, RNA polymerases, and distinct membrane lipids, reveals a closer evolutionary kinship to eukaryotes than to bacteria. Archaea are renowned for their ability to thrive in extreme environments, earning them the moniker extremophiles. We find them in boiling hot springs, in intensely saline lakes, in the frigid depths of the ocean, and within anaerobic environments where they produce methane, methanogens. Their unique biochemistry allows them to withstand conditions that would be lethal to most other life forms, a testament to their ancient resilience and distinct evolutionary path. They are the silent, enduring survivors, pushing the boundaries of what life can endure. And then there is eukarya, the domain to which we belong, along with all multicellular life forms, protists, fungi, and plants. Characterized by their complex, compartmentalized cells, true nuclei, and membrane-bound organelles, eukaryotes represent a later evolutionary innovation, profoundly shaped by those ancient endosymbiotic events we discussed. While fundamentally distinct from bacteria and archaea in their cellular organization, their deep molecular roots connect them to both, reminding us of the singular origin of all life. This grand tree of life with its three majestic branches illustrates the profound impact of genetic change and evolutionary divergence. Each domain, with its unique adaptations and biochemical signatures, represents billions of years of independent evolution, yet all are united by the common thread of DNA, RNA, and proteins speaking the universal language of life. But our exploration of genetic architects would be incomplete without acknowledging the most enigmatic and often maligned entities in the microbial world: viruses. Are they alive? This question has long been debated, for viruses are not soil cells. They lack the cellular machinery for metabolism and reproduction, existing instead as genetic material, DNA or RNA, encased in a protective protein coat, sometimes further enveloped by a lipid membrane. They are, in essence, obligate intracellular parasites, genetic elements in search of a host, utterly dependent on the cells they infect to replicate. When a virus encounters a suitable host cell, it attaches, injects its genetic material, and hijacks the host's cellular machinery, its ribosomes, enzymes, energy sources to produce new viral components. These components then self-assemble into new virus particles, which are ultimately released, often destroying the host cell in the process, to seek out new victims. It is a brutal, efficient cycle of replication, a testament to minimalism. Viruses, though not cellular, are powerful agents of genetic change and evolution. They can carry genes from one host to another, influencing the genetic makeup of populations. They are responsible for a vast array of diseases, from the common cold to devastating pandemics, but they also play crucial ecological roles, regulating microbial populations in oceans and soils. They are, in a paradoxical sense, both destroyers and shapers, driving the constant evolutionary dance of life on Earth. Their very existence forces us to expand our definition of life's influence, reminding us that the boundary between living and non-living can be surprisingly fluid at the molecular scale. As we conclude this chapter, dear friend, take a moment to reflect on the ceaseless dynamism of the microbial world. From the subtle shifts of mutation to the rapid exchanges of horizontal gene transfer, from the profound divergence into 3 domains to the enigmatic influence of viruses, life is a story of constant adaptation and evolution. The invisible architects are not static builders, they are relentless innovators, perpetually rewriting their blueprints, ensuring that the symphony of life continues in all its wondrous and diverse forms, echoing through the eons. We have truly only begun to listen to its ancient evolving melody.